Coming from my hometown, as well as Fali's hometown, who's made a mark for herself in the industry. She's the founder and current chairman and managing director of the biotechnology company called Avastha Jain Limited. She obtained a Master's of Science degree from the University at Mumbai in medical biochemistry and a PhD from IBMP, University of Strasbourg, France. Prior to a PhD, she worked as a research assistant at ICRISAT. This is the International Crop Research Institute for the Semi and Arid Tropics for 10 years. After her PhD, she returned to India and then followed it up with postdoctoral work at the University of Ghent, I presume, at a Rockefeller Fellowship and visiting scientist and Rockefeller at NCBS TIFR. TIFR is Tata Institute of Fundamental Research. Before founding the company Avastha Jain Limited, she, for a long term, created a strategic model to change the world and make it a better place with affordable healthcare and environment adjustment crops. The company, based on convergence of food, pharma, population, genetics, leading to proactive, preventive, personal and healthcare and food security, was unique in itself. She is the recipient of many awards, ladies and gentlemen, and titles too. The most important being Charles de Gaulle National Order of Merit, called the Lay Officer de Merit, Government of France, awarded by the President of France, Nicolas Sarkozy, in 2008. She has two daughters. Both are PhDs, very accomplished, from the University of Cambridge, accomplished scientists and businesswomen. Ladies and gentlemen, I present to you Dr. Villoo Morawala Patell.
Pleasure to be here, and also an honor to speak in front of the World Zoroastrian Chamber of Commerce. It's been and introduce you to the journey of Avastha Jain Limited. So just to say that the whole thing was based on the fact that India needed an innovation company. When I returned back with all those degrees, I went around India, visited universities, academia, as well as corporates and companies. And what I found was that while academia was fundamentally repeating the postdocs that they did overseas, good scientists, great people, but not inventing, not looking at what the world needs or what India needs. And in the same manner, if you looked at the companies, and they were all into generics business, and although they've improved considerably today, those days where it was a sad story to say. So I felt there was a need to create a bridge between academia and industry in India. And how do you go about that? And I had just returned, it was 95, I was in Sikandrabad, Hyderabad, and so Anji Reddy, Dr. Anji Reddy, who was the founder of Dr. Reddy's lab, met me, and he used to chat a bit. And he said, you know what, people like you go to Bangalore, Bangalore may provide you some space. So just picking up 2 suitcases and 2 children, I arrived in Bangalore 95 January, and with 2 chairs and maybe 35,000 rupees in the bank. And the rest is history, in the sense that I went to TIFR NCBS. They had just spun out the National Center for Biological Sciences, and asked them to give me some space. And that's where Avastha Jain really took root as an individual visiting scientist funded by Rockefeller Foundation. It was only in 1999-2000 that I spun out, I registered actually 2 organizations, Avastha Jain Limited, a profit-making company, and Avastha Jain Fund, a trust for non-profitable work. So sort of I was trying to create a public-private approach, but that just didn't work. And it was too early for its time. And so I said, okay, I'll take the route where I get my first term sheet. So ICICI Ventures were the first people who entered Avastha Jain with an investment along with the Tatas of €2 million. This was 2001. It was the first pure R&D company to be funded in India, or based on the concept that biodiversity, answers lie in biodiversity. Already most answers exist. And using the modern tools of genomics, proteomics, transcriptomics, and metabolomics, combined with bioinformatics, we could elucidate the story and recreate the world for, deliver both healthcare, prediction, prevention, personalization, and food security. It was a very ambitious play, and but was based on just a boat with 3 sails, with which I explained the business plan to these investors. The 3 sails were bacteria, human, and plants, and the hull of the boat carried the technologies. And from those, so I created horizontal platforms because I came from the field of genomics, I created the horizontal scientific platforms, and from that, today, has emerged a pipeline of technologies. Today, Avestagen is in the last 18 years of its existence, has raised almost 400 crores in equity, pure equity. So we have investors like ICICI Ventures, Tata's, Godrej, Cipla, Fidelity, New York Life. So I was able to raise purely on the concept of innovation. And from those innovation pipelines, around 2008, we already had a pipeline in pharmaceutical, in nutrition, in agriculture, and diagnostics. Unfortunately, the IPO intervened, and that has delayed us. Today, the company is poised to deliver all these products into the market. In the meantime, we began the Avesta Genome Project, which was, it was very important. So while the first phase was based on biodiversity as a basis of innovation, the Avesta Genome, in 2007-8, it became, something was telling me it's imperative to start this project. You were, you were working, your population was dwindling, the Parsi population, and I had all the technologies. I felt it was a perfect marriage to bring together these two. So today I'm going to focus my talk not on Avestagen of yore, which has now about 40 to 50 products getting ready for the market through 4 subsidiaries, Ava Seeds, Avesta Nordic, Avesta Good Earth Foods, and Aquas. Those are the 4 100% owned subsidiaries of Avestagen, which will take the research of the last 15 to 18 years towards the market, and we are launching them one by one. I'm focusing this on the next phase of Avestagen. What Avestagen is fundamentally a R&D company. It's an innovative company, and how is it going to innovate? But we were always ahead of time, and so Avesta Genome Project, when it was conceptualized, there was no one in India, and there was only Decode in the world looking at genomes as a basis of precision medicine. So Avesta Genome Project is a very ambitious project. It's built on very deep scientific foundation, and the aim is to provide global cure through precision medicine. And so the first step was to conserve the biological heritage of this community that came to India. As you all know, we are all part of it. They came to India around 12 to 13 centuries ago, and they have, in fact, and then they came to India is a phrase I've created to show that how much the community will end up eventually contributing to this project, not only to the nation, not only to its members, but to the world. So I think I would, with respect to time management, I think all of us know that we landed in Gujarat in the 8th centuries. We are largely an endogamous and non-proselytizing community, and we have maintained unique customs and food habits. This is extremely important data when you're doing such projects. Your social habits and all of that become very integral to the outcome of this project. And we are very well documented. We are a closed-knit community, and genealogical charts are available. So based on all this, we decided to create this project. This was 2007 to 2008, this project was conceptualized. And this is a community that went from refugees to nation builders, and surely they would have certain traits and certain genetic predispositions combined with their longevity that would, and the endogamous nature of the community that could result in identifying unique markers that would deliver precision medicine. So this is a bit, this is a quote from Fazana and Parsiana, and it just shows that the demographics reflect a vanishing community, and the deaths far outweigh the birth, and the females and males marrying out of the community. Now that is a question I'm not even going to hold here, because we believe we are on the threshold of change now, even in this, and maybe the numbers will increase, who knows. And there's the Geo-Parsi scheme. So preserving the unique biological heritage acquires the most importance in light of the rapidly declining population of the Parsi community, and perhaps to use this as a contribution for global healthcare. So I mean, this is rapidly decreasing numbers, as I've already said. It is an ideal population to study the interplay of multiple genes, environment and health. So what we did was we collected, I mean, I'll come to the collection later, but this is some of the data we found, is that we had higher incidence of stroke, Parkinson's, breast cancer, prostate cancer, and so the objectives of the study were defined as conservation, which was very important, conservation of the biological heritage of this endogamous community, building an archive of biological samples from 25,000 individuals, along with the genealogical and medical histories and social and dietary habits. A 65-page dossier, questionnaire was made, which some of you here in the community also wrote out, because we did these collections. Whole genome sequencing of 25,000 Parsi individuals would be done. Integration of this data in a systems biology manner, because it's not only genomes, but we are also looking at proteomes, like protein expression in the cell at that time of collection in the blood, and the transcriptome and the metome. It's a very deeply layered study, and the focus will be on cancers, neurodegenerative conditions, metabolic disorders, and autoimmune conditions. So we developed state-of-the-art platforms for conducting these parallel studies of genome, transcriptome, proteome, metabolome. And we created disease-specific family pluripotent stem cell lines. So blood was also collected. Some of you who were part of the collection will remember that there were 9 tubes per human being taken of 7 milliliters each, and each tube was dedicated to a particular purpose and a particular platform. And the outcome of the pluripotent cells means your stem cells will be generated from the bloodlines that will be kept in storage and could be used at any point of time. The Parsi community will be directly benefiting, and we said that the Parsi community would directly benefit through better understanding of the causal genes and other factors causing the diseases prevalent in the community. And we said any outcome and any drug or marker or diagnostic outcomes of this work would be given free to the community on the completion of the work, or because this is a work in progress and data comes out, and then you have to go through the whole cycle towards clinical trials and then to the market as a product. So we have a very powerful scientific board. Rita Caldwell, she's the chairman of our board. She's a professor at NIH, as well as she was an advisor in both Obama and Clinton administrations. And it's a very, very well-known board. I won't take you all through it. So this is a systems biology approach. Just a schematic arrangement of how we carry out the work, blood collection, archival. PBMCs are taken off immediately post blood collection, because each platform behaves in its own direction. you have to do immediate restoration or analysis or extractions as the collection happens. And so as you can see, once it's gone through, we've understood each human being entirely, because you have 65 pages of social data combined with all the data from each individual. And so you basically outcomes would be precision medicine and preventive medicine in this case, what you call nutrigenomics, and disease-specific stem cell lines. These are the three major outcomes as a first line. So Avasajan basically spent, I won't say burnt, 15 crores on this whole strategy and collection. And the collection of our samples was across India. Hyderabad and Secunderabad, we collected 430. It is all voluntary, and the ethical committee goes through it. It's under the Ethics Committee of the Government of India, all of that. So it's done absolutely with proper understanding. Navsari, we collected 687, Surat 708, Ahmedabad 560, Pune 513, Bangalore 178, and Mumbai 1108. And I can tell you each of these exercises is worth a book. I mean, actually I want to bring out a coffee table book about what happened while we were collecting, and it was quite an interesting, shall we say, journey, adventure. And Chennai, Kunur and Delhi. And so altogether we have collected so far 4,437 samples in 10 different locations between 2007 and 2012. And from our collected blood, we see this such a distribution of the diseases in our database, and cardiovascular system disorders being the maximum, and cancers, followed by cancers, and then musculoskeletal disorders, we found quite a number, and neurological and psychiatric disorders, etc., and others. In the cancer, we found a large chunk of breast cancer people, and gynecological disorders, digestive system cancers, head and neck cancer, blood cancers, etc. So it's a queryable database, so we can combine. We can question, I want a group of patients that have breast cancer but also have diabetes, and you can pick that group out and start your work on that, mining them, deep mining them through the genome-proteome process. So it's a totally Make in India project. It was built here, conceptualized here, and is going to grow from here. And the project would join some of the major global genome projects to understand human diseases and additional uniqueness on this endogamous community. And it will be conserving all our biological resources for time to come, because 2 tubes are not going to be touched ever. They are frozen for life. And so it's many of the genome projects around the world are not looking at it the way we are. The systems biology approach and the big data analysis. So ultimately we will understand novel disease pathways and biomarkers linked to those pathways. Oops. And the causative mutations and the resources developed here would be useful for generations of scientists. This is not something for the next 5 years. This resource can be mined for the next 100 years. So it is an extremely important project for the community to back it and take it to the next level. At this moment, I'm sort of commercializing my work of the last 15 years, but my work going forward, after I build up the CEO lineup for those the new commercial companies, I'm going to focus on this for the next 10 years to make sure that it sees the light of the day, and we at least deliver 10 novel pharmaceuticals in India. Because to date, even 65 years later, we have not been able to invent one single novel pharmaceutical in our country. So this project will do that. Besides developing skill development and of thousands of students and scientists. The story so far, as I've already said, we have collected and archived the blood samples. The database is building and it's continuing to build because there's a lot to go there. And we have so far invested 15 crore of our Vestigen's money into this project, and equipments are there in order to demonstrate the power of systems biology to understand diseases at the holistic level. We decided to prove that this model works. We took breast cancer for a pilot study, and as it has higher prevalence in Parsis and is globally the most common cancer in women. And we've completed the whole genome sequencing of 11 genomes, and 3 of which are familial in nature, meaning they're family inherited, and others are sporadic, and with age match controls. And the differences we have seen compared we have seen interesting outcomes at the level of proteome, where 17 differentially expressed proteins, including 7 novel zinc finger proteins, were found. Now these become a basis for drug discovery. So in these samples, we found unique zinc finger proteins, which could be further taken up for drug discovery. And in the metabolome analysis, we found again 23 breast cancer specific metabolites. That's a signature. And maybe this is the first clinical trial we will do with our friend Farzan Engineer, and we will take this signature and check it out if it is present also in other breast cancer patients. And we found this unique metabolites. And of course, I won't go here. Clearly, we did a HapMap population study also to see where does this population lie from the sequencing we had carried out. And the Parsis, first of all, I want to say Parsis are a unique group. They don't line up with anyone really, and from our data. And if at the nearest population is the Caucasian population, but even against the Caucasian, the Zoroastrian population stands alone and unique. This was done with Professor David Reich at Harvard Medical School. We did this together. And as you can see, the green is standing away from the other populations. The green dots that you see is the Zoroastrian population, and it is special. Just to take you through some of the differentially expressed proteins, this is data and slightly in gray because we don't want you all to know that this is being patented. I don't want anybody to call, that's for others, I'm joking. But these are the new discoveries for the breast cancer. And these are the metabolome markers, the ones which are lit up in red, which will be the signature for the early diagnosis. Even before the onset of the disease, we could look at that signature. And so these are the elevated metabolite levels in plasma. And the work was published in Genome Biology, and also in Nature it was reported, and in many other publications we were there, vetted out. The method of extraction and separation of the metabolite biomarkers was unique, and it was patented, and this was reported in Nature Biotechnology. You have to enlarge that little block there. So, we've done so we are very well placed there for those biomarkers in terms of patent position. And so now what next? So the focus now for the next 10 years, starting 2018, would be to create a Systems Biology Institute for endogamous population of India, and we are raising 399 crores over a period of 5 years to complete this piece of work. I'm just going to cut short, not going to read out all of it. So we will be collecting 25,000 Parsi individuals, and I'm in the midst of putting together the plan for raising this funding from both trusts and private equity too, or alliances, strategic alliances with companies. So I've just put together the timelines, what would happen across the overall work plan has been built, and the fallouts have been, anybody wants to have a detailed look can ask me, but I'm not going to consume time. So by the year 5, you would have finished the job on 25,000. You would have data pouring out, and you would be, handling big data of multiple platforms, and also by bringing focus on 4 disease areas, there will be outcomes of it. Although it can be applied, that data can go into multiple other diseases too, but we would bring outcomes out in those 4 verticals. And who are our competitors? I mean, who else is doing it? I mean, there's the Icelandic population study by Decode, which was only genome based, but Amgen just acquired, not just, but 3 years ago acquired it for 450 million just for the data, and today Amgen's full pipeline is based on this. I mean, the future pipeline. There's a 1,000 genome project. Carried out by US and EU. It's a 120 million project, 120 million. And there's the 100,000 by the UK government, which is a 430 million. There's a million genome database from Human Longevity, which is Craig Venter's company, which is also, we don't know how much they are burning. Million Genome Project is 250 million, which is US government. And then there's the Avesta Genome Project, where we are saying 25,000 people of endogamous Parsis in phase 1. We need 60 million, which amounts to, as I said, 399 crores. And the breakup is here. And it's a building of an institution, and I'm going to attend that starting January 1st, 2018. Initial work was done in this collection, and much work ahead, and but that's what we have to do. And as we said, and I think not only will the Parsi community contribute to global medicine from its genetic pool, as Mahatma Gandhi said, in numbers Parsis are beneath content, but in contribution beyond compare, and we can perhaps prove him right. Thank you.
Much, Villoo. I'm sure you understood all the scientific terms. That's very good. If I can get 2 or 3 of you to kindly repeat what she said, it would be very nice. All right. But as far as I'm concerned, there are 2 things that came to my mind. One is the 15 crores that they have spent. One is the 299 crores that they will, 399 crores that she's going to raise. And for all I know, she sucked a lot of Parsi blood. From various parts of India she sucked. And when they tested it in the laboratory, they found traces of dhansak, they found traces of curry chawal, they found traces of patio, and all that sort of thing. So they're still trying to analyze what the hell is happening with these Parsis. So except for the genomes, they found everything else. Villoo, thank you very, very much. That was very scientific presentation. I mean, it was as high as the planes that are flying over us. But you're doing some great, wonderful jobs. And this morning I had also announced that she has been a great patron of the WZCC with the highest donation that she has committed to. Ladies and gentlemen, put your hands together for Villoo Morawala Patell. And may I request Farida Shroff to kindly felicitate Villoo.